Hi, my name is Elena Lazarus, and I'm very excited to be sharing with you today my study in the diversity and biosynthesis of resin glycosides in morning glories. It's estimated that there are over 1 million plant metabolites. These include both primary and specialized. The difference between the two is that primary metabolites are involved in the development, growth, and reproduction of plants, while specialized metabolites seem to have more of a function with given the environmental conditions for evolutionary advantages. Some examples of functions of specialized metabolites are abiotic and biotic stress responses, beneficial insect interactions, and communication between other plants. So we are interested in the morning glory family, which is involved in, our annual plants in the Convolvulaceae family. They are native to Mexico and Central America and have historically been cultivated for medicinal and decorative purposes. One of the medicinal compounds in morning glories are resin glycosides, which also have been shown to be allelopathic, which give them both a human and an agricultural value. So this is a generalized structure of, This is the generalized structure of resin glycoside, and there are two identifying features that we use to isolate and identify them during spectroscopy and analysis. The first is the sugar backbone, and the second is a fatty acid chain, which often forms a macrocyclic ring like this one here. So a little bit of background on what was going on in the Mogi lab before I joined. The figure on the left was put together by Alexandra Bennett, and what she did is she took a published phylogeny for the morning glory family, and she picked various different species from different genuses. Our goal is to look at the diversity of resin glycosides in an evolutionary context. One of our hypotheses is that there was one ancestor that first developed resin glycosides, and so potentially we are maybe going to see that the diversity increased as the species evolved. So what she did is she picked various species to represent as thoroughly as she could the spread of morning glories, which is a very diverse family. You can kind of see how it varies from the flowers we're used to seeing to plants that look a little strange. And Alexandra Bennett started to map the diversity of resin glycosides in both the root and the leaf tissue. As you can see from this figure, anything from one resin glycoside to hundreds per plant, depending on the tissue type and species. So one of my goals this summer was to confirm that the species she ordered based on this phylogenetic tree are actually the plants we're looking at, so our map of their diversity of the resin glycosides is true to the species we have. We ordered different seeds from various online dealers, including Amazon.com, and I'm sure as you're all familiar, sometimes things can be deceiving when you receive them in the mail. Our second goal is to study the biosynthesis of the resin glycosides in the morning glories. This is like a very early stage project, and so our first idea is for how are we going to study the biosynthesis? And what we've chosen to do is to try to establish a gene knockdown method. In other words, we're trying to turn off genes that we believe are involved in the production of resin glycosides, and if we are successful, and the genes are actually involved in that function, that we will not see the resin glycosides once we knock out those genes. So I spent my summer trying to establish the first ever virus-induced gene silencing method for morning glories. So this is an overview for how I went about building my phylogenetic tree to confirm that the species we had in our lab were what they came to us as labeled. Kind of the noteworthy features of this diagram are that we used 3 different methods to extract DNA, which I will discuss in the next slide. We then did a standardized, straightforward procedure for doing the analysis. We did PCR to amplify the barcoding loci MATK and TRNLF, which are highly conserved, well-researched barcoding loci, which makes them ideal for building phylogenetic trees. We then did PCR purification, Sanger sequencing, and we built the alignment using genus technology. So this is a brief overview of those 3 different DNA extraction methods I just mentioned. 2 of them are more novel. One is a very standardized. Our standardized method was a commercial kit, and our 2 more novel are called the 30-second DNA extraction and FTA card extraction. So there's 2 ways in which these 2 novel methods are similar. Those are the fact that they use a simplified process for extracting DNA, which uses a cellulose paper disc as a column and various different buffers. And the cards, the hole punches, are actually added directly to a PCR tube. The ways in which these two differ is the chemicals they use for the extraction and cleaning of the DNA. And, oh, sorry about that. What we actually found was that in our laboratory setting over this summer, the commercial kit was just highly effective, and so we did the majority of our sequencing and DNA extractions using the kit. However, we did have a couple of species where we didn't have enough DNA to use a kit for an extraction, and we were able to use the FDA card for those because they require significantly less plant material. And we were able to take this process all the way through sequencing and got good reads. So they are viable options for extraction. Now this is an overview of my phylogenetic tree, and it's kind of a lot to take in, so we're going to zoom in on the bottom. And kind of how we go about reading this tree is that the black sequences are sequences we did not sample ourselves. They come from NCBI to build a more robust tree, and also were used to confirm species identity. The colored lines are all DNA extractions I did myself. The blue are samples we were able to confirm on a genus and species level, meaning they were exactly what we thought they were. The red are samples we were able to confirm on the genus level, which makes us think that it is possible it's the right species, but we didn't have a 100% match. And then the green are samples that we believe came to us mislabeled because they were neither a genus or a species match, and we have ideas of what they actually are, which is what you see mapped here. Something kind of interesting about our tree, which threw me off at first, is that it looks like the Maramia genus, one of the types of morning glories, is split in two. What we actually found when we were researching how accurate our tree was is there's recently published literature that has suggested that the Maramia genus is being redefined and reclassified and has previously been off. So we found that our phylogenetic tree is actually well backed by published literature. Now that we have our, we've confirmed we have the seeds we think we have, we are moving on to the next step, which is starting to look at how are we going to establish what proteins are involved in the biosynthesis of these different compounds. And so we are establishing a method called virus-induced gene silencing. So the way we're doing this is we are taking a viral vector, and we've added our gene that we are using to, as our ideal methodology. We've chosen the PDS gene, which I will explain a bit more about in a minute. We take this gene, we use agrobacteria to do a bacterial transformation, and we have two different methods of infection, which I will show you after. Once the plant has been infected with this vector, the plant has a protein called a dicer that takes the viral RNA and cuts it up into readable templates, basically. And then there is a protein in the plant called the RISC protein, which uses this viral RNA as a template, and it starts to scan its transcriptome. Plants essentially recognize viral RNA as dangerous and does anything they can to prevent those sequences from being transcribed in the plant. So what the RISC gene does is it scans a transcriptome, and when it finds a matching template, it binds to it and destroys that part of the sequence. So what we've done by using the PDS gene, which is a gene that codes for an enzyme that is essential to the production of pigment molecules, we are tricking the plant into recognizing its own sequence as dangerous, therefore knocking it out. And when we would know we would have success when we would see a characteristic phenotype called photobleaching, which is like the lack of the production of these pigment compounds. This is an overview of how we did our infections. In this trial, our seeds were 3 weeks from germination to infection. The pictures I'm showing you here are doing what we call the infiltration method, in which you use a needleless syringe to inject bacterial solution through a stomata. You know your infection has been successful when you see this leaching, kind of spreading of the water throughout the plant leaves and more than just the needle point. Our other method of infection we called agro-drenching, where we simply poured a bacterial solution over the plants in the hope that some of our vectors got into the plant cells and into their transcriptome. It usually takes 1 to 2 weeks to show the phenotype. In this case, we harvested our plants after about 1.5 weeks. So this is an overview of all of the different methods and protocols we tried for virus-induced gene silencing. You might notice the only one that was successful was the first, which is okay. But we have been trying a variety of different, like I said, the 2 different infection methods. as well as 2 different strains of agrobacterium, various protocols for preparing the bacteria. And we also have tried infecting at various life stages. You might be asking yourself kind of like, why are we trying all of these different methods when we have one that may have worked? And as you can see in this picture, we do have the phenotype we're looking for, but unfortunately, the phenotype did not spread. The sign of an optimized infection is that the virus will actually spread throughout the plant, and you can see this phenotype as the plant grows. Because we didn't see that in our first trial, we are trying to optimize our methods. This is just a brief overview of how we went about confirming that our plant was a transformed phenotype. Starting on the far left, we have our first control, which was not infiltrated, no syringe was put to it, nothing was injected. Coming in one, we have our plant that was injected with the media that our bacteria was suspended in with no bacteria itself. Here is our PDS plant with the phenotype, and here is our last control where we took the same strain of bacteria with an empty vector and the same solution and injected that. Our purpose for doing this control was to prove that this phenotype was not just due to a bacterial infection generally, but actually the virus RNA we put into the bacteria. At this point, I'd like to kind of summarize what we did this summer. We were able to confirm genus identity for 25 of our 29 species. We were able to confirm on a species level 100% match 11 of those 29 samples, and we called into question 4 out of 29 of the samples labeling. This was a really important result because in the future, we are hoping to map our glycoside diversity to our evolutionary tree in the hope of seeing some trends or relationships where we can make some commentary on the resin glycoside production in an evolutionary context. So if we had misplaced these 4 species in both the wrong genus and species type, we would have completely skewed our results going forward. Initially, we established the first ever methodology for virus-induced gene silencing in morning glories. Like I said, never been done before, so we were very excited about that, even though it's still being optimized. And then additionally, we were able to confirm that the 30-second DNA extraction method, which was published just in 2017, and that FT8 card extractions are viable for use in the future. However, at this time, we believe they need to be optimized before being used in a robust research setting like ours. At this time, I'd like to thank Dr. Gaurav Mogi for welcoming me into his lab, Dr. Lars Cruz for mentoring me all summer, and the other members of my lab who were very supportive and helpful overall. And I'd also like to thank our funding sources. And at this time, I'd be happy to answer any questions. Yes?
Can you go back to the picture of your treatment?
This one? The one after. Oh, all right. Here we go.
So in that media bacterial vector,
Uh-huh.
it looks like there's a lot of necrosis in there. Is there a way to follow up with some gene expression assay to see if it is knocking down the target gene that you're looking for, or if that's just some general cell death around there, or is that totally?
Okay, yes. So we had the exact same concern, actually. Just for time's sake, I kind of breezed over it. The necrosis is obviously not optimal for a virus infection, so we are hoping to optimize our methods. To actually confirm it has the right plasmid, we are considering doing PCR for the template and kind of looking at like a DNA extraction of these leaves. Unfortunately, we didn't have time to kind of follow through with that, but in the future, that probably will be used to confirm the phenotype. Yeah?
So the white spots are actually phytobleaching, that's not just
No, so this is actually, well, we believe it's photobleaching because all of our plants showed the same characteristic phenotype, or all of these set. The only point of concern is the kind of black necrosis surrounding the photobleaching spot, which we need to kind of investigate a little bit more. Yes?
So it looks like in your base trials you tried different developmental timelines.
Mm-hmm.
Do you consider trying injecting into different parts of the plant as well, like different types of coddles?
Yes, yeah, and so we did try some of the coddlings, and in the future we're looking at maybe doing seed treatments. We're kind of messing around with different life stages, but that's definitely something we're exploring in the future.
So we should probably move on now.
Yeah, well, thank you so much.
You need to get on that.
I know.